Okay, we should be going live right now. I had some sound issues, but it looks like I've fixed them for the most part. All right, let's see how this goes. Good questions. All right, let's get started. So the first question I'm seeing is, how does polarity create hydrophilic and hydrophobic compounds? So let me go ahead and pull up those slides. Oh, sound problems are always delightful. I'm trying to see if I can switch over and still keep sound. Hold on. Okay, cool. Looks like I kept sound this time. So I'm going to go into chapter 2. And let's go into bonds and talking about bonds. Okay. So we're going to talk about covalent bonds first. And cool, it looks like my sound is working, so that's awesome. So let's take a look at these slides. So the first question we're addressing is, how does polarity affect hydrophilic or hydrophobic? Something I think that we have to go back to before we can talk about that is we have to talk about what a covalent bond is, and that's kind of our base here. Okay, so just a quick reminder that a covalent bond is when we're sharing electrons. Okay, so a covalent bond is when we share electrons, and here we have a hydrogen atom and another hydrogen atom sharing electrons right here in the middle. And then here we have oxygen atoms sharing 2 pairs of electrons. We've got nitrogen atoms sharing 3 pairs of electrons. Okay, and these are regular covalent bonds. Okay, these are non-polar covalent bonds. Polar covalent bonds look a little bit more like this. So for a polar covalent bond, we actually see this electron shell of the hydrogen actually gets stretched a little bit. These electrons get pulled a little closer to oxygen than they are to the hydrogen. And we get this little bit of positive, and we get this little bit of negative, and we get this little bit of positive. So we get this end of the oxygen that has this negative because it's pulling on these electrons. Okay. Polarity means the unequal sharing of electrons between atoms. There's an unequal sharing of electrons that's happening, and the unequal sharing results in a partial charge. The unequal sharing results in this area being negative and this area being positive. And this is a diagram of a water molecule itself. So this is what a water molecule looks like. So water molecules are inherently polar. They have this kind of negative area, they have this kind of positive area. And so that means that this water molecule is going to like other things that have either a little bit of polarity, okay, or things that are fully charged. So if we look at oxygen interacting with a hydrogen of another water molecule, what we'll see is a little bit of this negative getting attracted to this little bit of a positive, which is going to be pretty beneficial. That's going to keep all of our water molecules together. Okay, so it's going to keep them together, it's going to keep them closely bonded, it's going to make them attracted to each other because they are both polar. Okay, so things that like water are what we call hydrophilic. And things that like water are going to be able to interact with this polarity of the water. Okay, the main thing that determines if you like water or not has to do with if you have a charge or not. Let me see here. Water as a solvent. Here we go. Okay, so water as a solvent is water is really great at dissolving things. Okay, so a solute is something that you dissolve inside of a solvent, and water is a really fantastic solvent because it's going to dissolve anything that has a partial or a full charge. So here our example is we have sodium chloride. We actually have a positive, sorry, if there's screaming in the background, it's not a person, it's a cat. So we've got this positive sodium right here that's attracted to our negative oxygens here, and we've got this negative chloride that's attracted to these positive hydrogens. So sodium and chloride are going to be very attracted to water because they're fully charged, right? This is a cation, this is an anion. And they're going to form these hydration spheres that we talked about, where these water molecules actually are going to surround these ions. We're also going to see things that don't have a full charge, right? This sodium has a full charge, this chlorine has a full negative charge, but here we've got glucose. And glucose is made of carbon and hydrogen, and it's also got oxygen. And as we know, oxygen is very hungry for electrons. So the oxygens on this glucose are going to be actually making this glucose a little bit polar. That's why sugar can dissolve in water, because it's got a little bit of polarity in some parts of the molecule. And here we can actually see our water molecules sticking on to those little bit of little spots of polarity in that glucose. Okay, so things that are hydrophilic, like water, because they are polar or charged. Things that are not charged at all, even a little bit, things like lipids, are not going to like water. Okay, so triglycerides here, we can see this big long chain of carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, all the way down. Carbon and hydrogen are very good at sharing electrons. And because they're good at sharing electrons, they're not polar. They're not polar, they're not charged, which means these are hydrophobic. Okay? The basis of something being hydrophilic or hydrophobic is if it is polar or has a charge, it will be hydrophilic. Okay? It will like water because water molecules themselves have a slight charge. Versus if you're hydrophobic, that means that you don't have any charge, you don't have a full charge, you don't have a partial charge, you have no charge at all, like this fatty acid right here. Okay, there's no charge, there's no area where there's unequal sharing of electrons. All the electrons are being shared equally, so we don't have any areas of polarity. Okay, so that's the first one. And I do have a slight guest appearance. This is the one who's screaming. I'm hopefully going to get her to stop, but she just really wanted to say hi. So that's the first question of hydrophilic, hydrophobic. We've also got hydrolysis and synthesis of ATP. Okay, cool. Let me pull those up. This is the end of our chapter 2 slides. Okay, so back into our chapter 2 slides. ATP, the function of ATP, the reason that we have ATP is so that we can temporarily store energy. So we're going to be able to store energy temporarily in our cells as ATP. And the reason that we can do that is because we've got these 3 phosphate groups over here. Okay, and that's where our synthesis of ATP, the synthesis and hydrolysis of ATP is actually going to happen, is with these phosphate groups. So let me really quick, that's kind of helpful. Let me really quick pull up a picture of ATP and ADP to help make it a little bit more clear. Oh, beautiful. Found exactly the picture I need. Okay, so hopefully all of you can see this. So this is another different diagram of ATP. Okay, so over here we've got ATP and here we've got ADP. So adenosine triphosphate, the reason that we show this as being the energized version, right, we show this as the one with the full battery, there's this bond here that has been created because we have 3 phosphates here, 1, 2, 3. We've shoved this 3rd phosphate on here, which is pretty tough to do. These phosphates all have a full negative charge on them, and they don't like being next to each other. So this is kind of like compressing a spring. Shoving this 3rd phosphate on here is like pushing a spring together. It's going to take some energy to push it together, but then it's going to be able to hold. We're going to be able to store that energy in that coiled spring. We're going to be able to store that energy in this bond. So they're going to be covalently bonded, but this bond has a lot of energy in it. And when we break that bond, as shown here, we end up with ADP, okay, diphosphate. We've only got 2 phosphate groups. So we pop off this outside phosphate, and we release that energy, usually next to something that we want to, build. We can release that energy and do anabolic reactions. We can release that energy and build proteins. We can do all kinds of things with that energy. We can do whatever we want in the cell with that energy. We can do transport. We can do building things. We can actually move proteins. We can move muscles. Okay, it's all coming from ATP and ADP. The synthesis of ATP is the reverse of this process, okay? So if we have these two phosphates over here, the synthesis of ATP is going to be shoving this phosphate back on here. Okay, so the synthesis of this is going to be shoving phosphate back onto here to get this high energy bond. And we're going to be able to do that when we harvest the energy from catabolic reactions. Okay, so when we break things down, we're going to release energy from the food that we eat. And we're going to use that energy to push this phosphate back on to this ADP subunit. Then we're going to store that energy as ATP and let that ATP go to wherever it needs to go to then use that energy. And that's the hydrolysis of ATP. When we go from ATP to ADP, that is hydrolysis. We're breaking this bond and we're releasing the energy. The synthesis of ATP is the other way. Okay, so the hydrolysis is going this way and the synthesis is going this way. Okay, so when we have hydrolysis, we're breaking a bond. Hydro, water, lysis, to break. So we're breaking with water. We're breaking this bond going this way. And then we're building the bond going this way. Okay, that's the synthesis of ATP. So when we have this, kind of back to the slides and what we were talking about here, the hydrolysis of ATP, when we hydrolyze this last phosphate group, that's going to release energy. We're going to go from our high energy form to our low energy form. And when we synthesize ATP, we're going to go from our low energy form to our high energy form. So we actually have an enzyme called ATP synthase that catalyzes the reaction to shove another phosphate onto this ADP. And if you've studied cell respiration recently, you'll know that the energy from one glucose molecule is used to we can make about 36 to 38 molecules of ATP for one glucose. Okay, just one glucose molecule gives us 36 to 38 molecules of ATP, which is a good thing because your body is using, millions of ATP per second. It's constantly being hydrolyzed and then synthesized and hydrolyzed and then synthesized just over and over and over again. Okay, so it's a very, very cyclical relationship between these two processes. Oh, another good question. Can you explain what an amino acid is and how that makes it different from other amino acids? Yes, I can. Let me pull up another picture. Okay. So we're going to look at some actual amino acids. Of course, now that I pulled up this picture, I realize this is not the one that I want. I want them to have the top part. This one's best. Oh, and it's on a site called BioNinja. Perfect. Okay, so when we're talking about amino acids, we're always going to see this top part here. Okay, this carbon, nitrogen over here, hydrogen, COOH over there. Okay, we're always going to have that top part. And then it's whatever's on the bottom is going to actually give it its identity. So we have a total of 20 amino acids, and those are all shown here. You don't have to know these, you don't have to memorize them. But here we can see glycine is an amino acid that has just a hydrogen. And then tryptophan is an amino acid that actually has two ring structures coming off of the top part of the amino acid. Over here we've got histidine, which also has a ring structure. We've got tyrosine, serine, asparagine, aspartic acid, glutamic acid. And we've got 20 different kinds. Some are nonpolar, some are polar, some have a positive charge, and some have a negative charge. Okay, some are really big, some are really small, okay, some are kind of in between. And those amino acids are going to determine the identity of the primary structure of a protein. Okay. So when we talked about amino acids, we kind of just looked at this very general structure of, okay, we've got an amino group, we've got a hydrogen, we've got a carboxylic acid group, and that's going to be the top part. And this R is going to be what changes. And so here on our example on the slides, we have glycine and alanine, where this R group is either a hydrogen or it's a methyl group. And if it's a hydrogen, that means it's glycine. If it's a methyl group, that means it's alanine. But that's not really the important part. The identity of the amino acid and kind of how those R groups work isn't the big takeaway for the class. What our big takeaway is that we've got different levels of structural organization. So the primary sequence of a protein is the unique sequence of amino acids. So what amino acids did you use and what order did you put them in is going to be the primary sequence. Then we get into actual folding of the protein, and that's where we start to see function. So here, our primary structure is which amino acids did you use and what order did they go in. Because you can imagine for some proteins, for some polypeptide chains, they've got hundreds or thousands of amino acids. And you're going to have a very different protein if you go glycine, glycine, alanine, versus if you go lysine, lysine, arginine, or tyrosine, asparagine, tryptophan. You can do any order of any of these. Okay, these are kind of like words in the human language. We can put a bunch of words in a bunch of different orders to have a bunch of different sentences. But then it kind of stops there, because once we make a sentence, you know, in the English language, we're kind of done. But for proteins, we're not. They string all of these amino acids together, and that's just the starting point. It gets really interesting once we make this primary structure of all these amino acids in the chain. Then we start to form the secondary structure, either helixes or sheets. And these structures are formed with hydrogen bonds, these little dots. Okay? That's a hydrogen bond. The bonds between these two amino acids, this is a covalent bond. We specifically call it a peptide bond. This is a covalent bond. It's going to be very, very hard to break. Okay? Covalent bonds are much harder to break than hydrogen bonds. So we have these very strong bonds in our primary structure. We're going to have these weaker bonds making up our secondary structure, and then those bonds are also going to help us make our tertiary structure, as well as potentially our quaternary structure. So our secondary structure, we've got these helices and these sheets, and then we take those helices and sheets and fold them upon themselves, and that gives us the tertiary structure. This is when a protein actually becomes functional. And what's happening here is these helixes and these sheets are actually going to further bond with each other, usually through hydrogen bonds, okay, or ionic bonds, but somewhat weak bonds, are going to hold this structure together. And then sometimes we have to put a bunch of these structures together, a bunch of tertiary structures together to get a quaternary structure for a functional protein. Which means if we heat up a protein, okay, if we heat up a protein or if proteins get too hot, the bonds that are going to break are the ones in the secondary, tertiary, and quaternary structure. If we heat up a protein, we're going to keep the primary structure, but we're going to denature the part that makes it functional. And that's because if you apply enough heat, you can break bonds. And we're familiar with this. If you apply enough heat to a pot of water, it's going to start boiling and it's going to start turning into steam. And to do that, you're breaking the hydrogen bonds between the water molecules when you make steam. But the water molecules themselves are still covalently bonded, okay, within that molecule. And it's the same thing here. As we denature a protein, we keep this primary structure, but we lose the secondary, tertiary, and quaternary structure. Okay? So for protein denaturation, hostile environments like heat, as I just said, you can also denature with acid or salt. That'll change the 3D shape, okay? And that will destroy its ability to function, because the function of a protein comes from its 3D shape. Okay, so we've got some idea what amino acids are, but our really big take home here is this primary, secondary, tertiary structure idea, in that these bonds are harder to break. So you're going to keep this order, but this folding you can actually undo, and you can denature. It's relatively easy to denature a protein. If you ever cooked an egg, when the egg whites go from being clear to white, that is denaturing a protein. Those proteins in the egg whites are getting denatured by heat. It's one of the reasons that it's so dangerous when humans get fevers, because if you heat your body up too high, your proteins can start to denature. Okay? They're very sensitive to their environment. Okay, so I've got a request to go over endochondral ossification. No problem. There's also a note of in the chemistry part, I said to review the formula of 2n square. and what that describes. I'm not sure what you're referring to, so if you can give me a little bit more specifics on that, Katrina, I will get back to your question once I go over endochondral ossification. But first, endochondral ossification. Okay, endochondral ossification. So when we're talking about ossification, hey, we're going to remember we've got two types. And when we're talking about ossification, we're now talking about bones. So we're talking about how bones get formed. So we're talking about the process of bone formation. Hey, how do we form bones? And we're going to note that we actually initiate this during the fetal stage, and we might continue into adulthood. Hey, we're going to actually initiate it when the babies are first growing. Bones can form in two ways. They can form by replacing connective tissue and by replacing mesenchyme in the fetus. So when we replace cartilage with bone, we're going to get endochondral ossification. Okay? Endochondral inside of cartilage, that's going to be the cartilage base type of ossification. So for endochondral ossification, we're going to start with a cartilage model. So we're going to have this bone here, okay, that's going to be a long bone. Okay, we see this in all of our long bones. And the mesenchymal cells that are in the baby's body are going to transition from mesenchymal cells into being cartilage. Okay, so this whole bone is made of hyaline cartilage, and then we're going to see that hyaline cartilage start to change from cartilage to bone. So in this middle region, we're actually going to see a pH change, and that's what's going to cause calcification. The matrix in the center is going to harden. It's going to go from uncalcified to calcified, and the chondrocytes are going to die because we're going from cartilage to bone. So now we're not going to have chondrocytes. Now we're going to have osteocytes, okay, or osteoblasts or osteogenic cells. Okay, now we're going to have bone cells in here instead. And for primary ossification, I really love this illustration. It shows that when we're doing this primary ossification, we've got to have this blood vessel here. Okay, so we're going to start in the middle of the cartilage model, and we're going to transition to bone tissue once we have this blood vessel come into the bone. Okay, so it has to have that blood vessel as a supply of for nutrients, as well as to take away waste in order to do this transition from cartilage to bone. And as we talked about as well with intramembranous ossification, the first type of bone that's going to form, we're going to form spongy bone first, and then compact bone is going to come later. So we're going to start in the middle of the bone, and we're going to go from cartilage to bone, kind of radiating from the center out. Okay, so we're going to start in the diaphysis, and we're going to go from the center of the diaphysis out towards the epiphyses. And we're going to change the matrix, which is cartilage, from cartilage to bone. First spongy bone, and then compact bone eventually. And then once we get to the end here, okay, this is our primary ossification center in the middle of our diaphysis. The middle of our diaphysis is going to have this primary ossification center, and it's going to spread out to the end. And once it gets to the end, we're actually going to have another ossification center form. And that is pretty important for a couple reasons. If we just kept going from our primary ossification center all the way out to the edge, we might miss a couple important remaining cartilage areas. Okay, there's some cartilage that needs to remain in the epiphysis of the bone. So our secondary ossification center actually forms in the middle of our epiphysis right here, okay, which is currently all cartilage. And again, our artery has to enter to be able to bring in nutrients. And we're going to see this expansion, kind of now a radial expansion, okay, kind of expanding in a circle from this secondary ossification center out. And the fact that we're going from the middle out is going to leave articular cartilage, okay, on the end of the bone, because that's where the bone is going to articulate with another bone. That's very important. But we could probably keep articular cartilage if we let the primary ossification center go all the way to the edge and then stop. We would still get articular cartilage. But if we let our primary ossification center go all the way to the edge of the bone, we would miss this layer, okay, of cartilage that remains. Our secondary ossification center kind of balloons out and it stops, and so it leaves this cartilage here on the end, but it also leaves this little layer of cartilage right in here. This is our epiphyseal plate. And our epiphyseal plate is actually our growth plate. This is where we're going to be able to keep lengthening the bone from, okay, is this plate of cartilage. This is where we're going to be able to get this kind of differentiation of cartilage to bone, and we're going to be able to keep growing and keep growing and keep growing those plates until we hit the size of the bone that person's body is going to eventually hit and stop. And then those plates will seal, and then usually by the age of, say usually by the time a person is about 21, 22, their epiphyseal plates are gone. Okay. Sometimes it can be later, sometimes it can be earlier. Okay, but once you stop growing, then your epiphyseal plates close and they're done, and your bones are done growing. Okay. But as a child, it's really important to have this epiphyseal plate so that your bones can continue to grow, so you can keep getting taller. So for endochondral ossification, we're going to get this central primary center, and then we're going to get the secondary ossification center, and both of them are going to form spongy bone first. Okay? And the spongy bone in our epiphysis is going to stay. Okay? It's going to stay here. We're not going to carve it out for the medullary cavity like we do in the diaphysis. We're going to keep the spongy bone in the epiphysis, and as it expands out, later we're going to form the compact bone on the edge, but we're always going to do spongy bone first. And the advantage of doing a primary and then a secondary ossification center is having primary and then secondary allows us to have that articular cartilage preserved on the end, as well as the epiphyseal plate of cartilage preserved in between the diaphysis and the epiphysis. Okay, got a question on the study guide. Let me see if I can pull that up. Give me 1 second to get a document open. Katrina, that is a fantastic question of what is that? And that is an editing error. It's an editing error on my part. I should have edited out that question. I reviewed the formula 2n square. What does this describe? We did talk about the one right before it on that study guide. What is atomic number? What is valence-valency? As well as what are electron shells, which is the question right after. Is we talked about atomic number and electron shells, but we didn't talk about the 2n square formula. That is something that can be talked about for chemistry, but with the way that we address things, we did not address that, and that was just an editing error. I didn't catch that to edit it out. Okay? So you don't need to be aware or worried of that. Okay. Calcitonin and PCH for bone remodeling. Cool. I can do that. Bone remodeling. Okay, I'm actually going to start right here. So calcitonin and PTH are two hormones that are involved with bone remodeling, because bones are constantly being built and then broken down a little bit, especially in areas of activity or stress. So the distal end of your femur is constantly getting remodeled, okay? But the shaft of your femur, not so much. So the main two players in this are going to be this hormone, calcitonin, as well as this hormone, PTH. And those are going to be involved with calcium specifically. So we do have quite a few kinds of salts and other things that we're storing in the bone, but the main one that we're going to focus on is calcium. Okay? A major salt that we deposit in the bones is calcium. And so with calcium, we're going to want to keep a homeostasis of calcium in our blood. And the reason why is because it's involved in a lot of really important things, like how your nerves function and how your muscle cells function and how your blood clots and how your enzymes function. So if there's changes in the blood levels of calcium, you can die. If it gets too high, you can go into cardiac arrest. If it's too low, you can go into respiratory arrest. So your body closely regulates this level of calcium between 9 and 11 milligrams. So I really like this picture. I really like this illustration of showing if our calcium goes above 11 mg per 100 milliliters. Okay, so if we get too much calcium, our body is going to respond. So there's going to be a way for our body to detect that our calcium has gotten too high. And if the calcium levels are too high, the thyroid gland is going to start secreting a hormone called calcitonin. And calcitonin stimulates the depositing of calcium. So the job of calcitonin is to take a message to the bones and say, hey, you need to have more osteoblast activity. You need to be depositing calcium. You need to be building more bone. Okay, so whatever you're doing, you need to build more bone. So it stops osteoclasts, okay, which are the things that degrade bone. So it stops osteoclasts and promotes osteoblasts so that we get the calcium out of the blood. The whole point is that we get too much calcium in the blood and we got to get it out of the blood. And the way we get it out of the blood is we take it from the blood and put it in the bones. So we put it in the bones through this hormone. Okay? This hormone means put more calcium in the bones. And that's what the cells will respond to do. The other side of things is if our blood levels of calcium drop too low. So if our blood levels of calcium get too low, our parathyroid glands are going to release this other hormone, PTH. And the job of PTH is to notice, okay, we've gone too low in our level of calcium. We need to bring it back up. So if we've gotten too low in our level of calcium and we need to bring it back up, we're going to need to break down some bone. So if we're bringing levels down, we're going to have to bring them back up. And the way that we do that is we stimulate osteoclasts to degrade the bone. We want to release the calcium that's in the bones. So we're trying to release the calcium that's in the bones. And the way that we do that is through this hormone. This hormone is going to be the one that carries the message to signal osteoclasts and say, hey, you guys need to step it up. You need to get more active. You need to break down more bone. We need more calcium in the blood ASAP. So if we get too low a level of calcium in the blood, we're going to secrete PTH. That's going to increase osteoclast activity, and we're going to actually pull calcium from the bones and put it in the blood. And the other side of things is if our levels of calcium get too high, we're going to want to pull those calcium ions from the blood and put them in the bone. And the hormone that signals to do that is calcitonin. Okay? Calcitonin is the signaling to say, hey, we need to take some of this calcium and put it from the blood into the bone. And for either one, we're going to keep doing this cycle until we hit the level that we need to be at. Okay, so this PTH is going to keep being produced until we get back to a level of 9 to 11 milligrams. And this calcitonin is going to stay active until we get back to this level of 9 to 11 milligrams, and then it's going to shut off. Okay, because once we're at the right level, if we're at the right level, we're not going to produce any ridiculous amounts of calcitonin or PTH because we don't need to do anything with the levels of calcium. Okay, if our levels are good, we're not going to mess with them. We're going to leave them where they are. If they get too high, we're going to produce calcitonin to put the calcium back in the bones. If they get too low, we're going to produce PTH to pull the calcium from the bones. Okie dokie. So that is calcitonin and PTH. And, oh, perfect timing, just when I needed another question. Mechanism of enzyme action and role of cofactors in an enzyme. Yes, I can talk about that. Give me 1 second to find those slides. All right, got it. Okay. enzyme action and role of cofactors. So the purpose of an enzyme is to speed up a chemical reaction. Okay, we're going to want to catalyze some chemical reactions and make them go a little bit faster. So the way that that happens is that we're actually going to have our substrate, okay, like sucrose and water in this example for our enzyme sucrase. Our substrates are going to bind to the enzyme, and then the enzyme is going to catalyze the reaction, usually by putting some physical stress on the bonds. So normally it's very hard to pull these two apart, but what the enzyme is going to do is it's going to kind of bend them so that there's a little bit more stress on that bond, so it's easier to kind of hit something at it and break it. It's then going to release the products, it's going to be empty, and then it can take in another substrate. So our enzymes are going to keep doing this. They're reusable. We can keep using them. So our enzymes can take in substrates and then build them up or break them down, and then spit them out, and then they're empty, and they can do it again. We did talk about cofactors, and there's a quick note that you might run into this term, apoenzyme. Okay, so an apoenzyme is another term you might run into to talk about the fact that apoenzymes are the part that's made of protein, but sometimes in the active site, sometimes in the active site of an enzyme, just the protein itself isn't enough. Sometimes we need something like a cofactor, and a cofactor is something that's in the enzyme, but it's not a protein. So sometimes in the active site of the enzyme, we'll find a metal ion. Oftentimes we'll find things like iron or zinc or magnesium or calcium, which we already talked about how important it is to regulate that in the body. Sometimes we actually use really big organic molecules derived from a vitamin. And that's just that sometimes enzymes on their own aren't enough to be able to catalyze the reaction. So we have a cofactor that will actually sit right in here. So we'll have like a zinc ion just kind of sitting in this pocket over here. And then when the substrate comes in, the zinc can help usually with the shuffling of electrons to make it so that the enzyme can function more easily. Okay, I think I touched on everything for enzymes. If there's still questions that you have for enzymes, let me know, because I think that touched on the two things that you wanted to know about it. And we also want to go over hydration spheres again. So we go up a little bit. So hydration spheres happen when we have something that we dissolve in water. And so a hydration sphere is going to be the surrounding of this charged particle by water molecules. And the surrounding of this charged particle by water molecules is going to be what we call a hydration sphere. And this is helpful because this can happen in the body. So in our body, we have these sodium ions and we have these chloride ions just kind of packed in with these little water molecules getting transported through our body. Okay? All of our electrolytes are going to be kind of bound up in these hydration spheres. A hydration sphere just refers to the fact that anything that has a partial or a full charge is going to have water molecules stuck all over it. Okay? And if you look at that little kind of, this whole conglomeration of molecules, this whole conglomeration of molecules, including the water molecules stuck on it, is called a hydration sphere. Oh, I just did the entire explanation on the wrong set of slides because I'm still showing the chapter 6 slides. Ha! Hydration spheres. Oh, I did enzymes on the wrong slides too. Okay, I'll really quickly go over all that again, just with a visual this time. Okay, so what I was just saying with hydration spheres is that here we have this negative chloride, okay, and we've got all these little waters around it, so this is a hydration sphere. This is a negative, so it's going to have all these water molecules stuck to it. And this is a sodium, it's fully positive, so it's going to have all these water molecules stuck to it. And that's going to make this little hydration sphere, okay, of all these water molecules stuck to this charged little particle. And that's going to allow for things that have a full or partial charge to be easily transported, usually through the blood, which is mostly made of water. The other point was enzymes. Okay, so our enzymes here, this will make a lot more sense with what I was saying earlier. So this is sucrase. Here we've got sucrose and water. Sucrose and water are going to enter our enzyme here. And then, as I was saying, our enzyme is going to put a little bit of pressure on that bond to then break it and release the products. Then our enzyme is empty, and we can then go back with our empty enzyme and pick up more substrate, and then break them apart, and then it's empty, and then it can pick up more substrate, and then break them apart, and then it's empty. And our cofactors, like zinc or magnesium or iron or calcium or vitamins, those cofactors are usually going to sit in this active site, okay? They're going to sit in this little pocket. And usually there's just one, okay? There's just one cofactor. So there'll be like one zinc ion or a calcium ion or a magnesium ion. Usually won't have a whole lot of other ions kind of packed in there. Okay? And they're just a little helper. They're called a cofactor because they're just there to usually hold on to electrons or just help out with the process as the enzyme grabs onto these substrates. And so that's the role of functionality of an enzyme, is that they're going to be very specific. Okay, so each enzyme is only going to act on one substrate. Okay, they're highly specific. Our substrate is going to bind to an active site, and the job of an enzyme is to speed up a reaction. Okay, so we're trying to make our reactions move more quickly, and enzymes are going to be the thing that lets us do that. And as I mentioned, our cofactors, we're going to either see ions or vitamins that are going to bind to it and help the enzyme activate, or help the enzyme then be active to break things down or build things up, depending on which enzyme it is. There's a question about two subtypes of connective tissue. And as far as a lecture exam, connective tissue is not really going to be focused on the lecture exam. Okay? It's more going to be focused on for the lab exam, okay? For knowing all the different kinds of connective tissue and their purpose and where you would find them. There might be some kind of more basic questions for things about connective tissue and epithelial tissue of, you see a tissue that looks like this, what might this be? Or here's a description of a tissue, what is that? But it's kind of for the bigger classifications of things like connective tissue and epithelial tissue. That's going to be for lab. Okay? So just kind of the basics of it. But as far as knowing the subtypes, that's not going to be important for the lecture exam. Okay. I'll hang out for another couple minutes and see if anybody else has any more questions. And if there are no more questions, then I will sign off for the night. And if you have any last minute questions, you can send me an email. I will do my best to get to your emails before the exam tomorrow at 3. Biochemistry section of the study guide. And just to be clear, Viviana, are you talking about what are the types of carbohydrates? What is the general formula for a carbohydrate? I know the 3 types of monosaccharides that can be absorbed by the human body. Cool. All right, so let's talk about carbohydrates. Which is going to be this one. All right. So carbohydrates, you are going to be able to recognize a carbohydrate because it's going to be formed by carbon, hydrogen, and oxygen. Okay, so usually we're going to see 1 carbon atom for each water molecule. So we're going to see usually about 6 carbons and 6 oxygens and 12 hydrogens. And the main job of a carbohydrate is going to be to produce energy, okay, but they can also sometimes be used for structural purposes. So we've got different sizes of carbohydrates. We've got small ones, we've got monosaccharides, we've got disaccharides, which are a little bigger, and then we've got polysaccharides, which are much bigger. Okay? And most of them are going to be hydrophilic. Most of them are going to really like water. So our monosaccharides are simple sugars. We're going to have usually about 6 carbon atoms, but it can range anywhere from 3 to 7. And there's 3 simple sugars that we can absorb in our small intestine without any other kind of digestion. And that's going to be glucose, fructose, and galactose. And glucose we usually find in syrup or honey, fructose we find in fruit, and galactose we oftentimes find in dairy products. Okay, so those are the 3 types of monosaccharides that we can absorb from the human body. General formula is going to be 1 carbon, 1 oxygen to 2 hydrogens. And then here's one type of carbohydrate. These are monosaccharides. Another type of carbohydrate is a disaccharide. And we make a disaccharide by combining 2 monosaccharides. So glucose and fructose yields sucrose. Okay? This sucrose, this is 2 monomers linked together. And then last but not least, if you have a lot of saccharides, monosaccharides linked together, we're going to get what we call a polysaccharide. So we're going to have tens or even hundreds of monosaccharides all joined up. So we've got this starch molecule, which is just a bunch of monosaccharides all attached to each other. And then glycogen, we have, again, a bunch of monosaccharides all attached to each other. And then cellulose is the same thing. The difference between these polysaccharides has to do with how branched they are. So starch is a little bit branched, glycogen has a lot of branching, and cellulose doesn't have any branching at all. So in animals, like in our cells, we're going to find glycogen mostly. We're going to be seeing glycogen in our bodies as a storage molecule in liver as well as in skeletal muscle. Glycogen is going to be a really fantastic storage molecule so that if our muscles have to break down energy really quick, right, if we're doing some kind of exercise, our muscles are going to really need energy really, really fast. So they're going to need to be able to get energy really quickly, and the easiest way to get energy is going to be to pop off one of these sugars and then use that for energy. Okay, so this is a great storage molecule for us. Starch is how plants store energy, and then cellulose is what gives them structure. So we can get starch and cellulose from our food. We can also get glycogen from our food, but if we're eating plants, we're going to be getting starch and we're going to be getting cellulose from our food. We can digest starch. We can digest starch and get energy from it. We cannot get energy from cellulose. Okay, this is like the chewy, stringy fibers of celery. We can't digest that. It just passes right through us. We can't actually break down the cellulose. There are some lower level herbivores that can break down cellulose. Things like cows, they have the right bacteria that can break down cellulose. And some insects can also break down cellulose, but we unfortunately cannot. Okie doke. Looks like that is all of the questions. I apologize for the minor technical difficulties that happened at the beginning. I believe I've got them fixed for next time. So if you have any other questions, feel free to email me. As I said, I'll try to get to you. I'll try to get back to you before 3 o'clock tomorrow. And the exam is going to have some multiple choice, so make sure you bring an AccuScan form. And then it's also going to have some free response as well. So with that, I will wish you all a good night, and I hope you all have good luck studying.